With single-cell RNA data, a substantial portion of the transcripts will lack sufficient expression in any of the cells or sufficient variance across the cells to be considered for further analysis. As a result, even though data is collected on over 30,000 transcripts, typically only 1,000 to 2,000 transcripts will have sufficient variance across the cells. So the first step in RNA data processing is to identify these genes with sufficient variance. In addition, mass will carry forward genes that have high expression even if only a few cells express these genes. You can start this process by clicking on Normalize here. Mass will automatically run the computation to identify HVGs and display this plot that you see here. Note that variance will end up with a biased set of genes primarily expressed in large cells and ignore several genes that are expressed and even differentially so in small cells. As shown in this plot, it is therefore best practice to select with the highest level of variance across the range of mean value. So usually we select the cells that are at the high end of the curve along the curve. In addition, mass will also include genes that may not have sufficient variance but are expressed in a few of this HEG plus and HVG plus bucket. You can also browse the list of genes here, and you can search for any genes that might be of interest to you based on the biology that you're studying and understand if any of those genes are not selected, and if you want to manually include them in further analysis, you can do so by simply clicking here. Once you're satisfied with the list of genes that you have selected, you can click Save to include for further analysis. Once the HVGs have been identified, the next step is to normalize these genes to account for differences in cell sizes. As we discussed earlier, large cells will contain a larger amount of RNA than the smaller cells. As a result, when conducting differential analysis, for instance, between cells of various types, the true biological difference will be obscured by the difference in background. As in the case of identifying HVGs, when normalizing, one must account for the dependence of variance on mean value. In mass, the recommended method of normalization is SC-transform as implemented in the Surat R package. You can run normalization by simply clicking on Run Normalization here. This process may take several minutes to complete. Once the normalization is completed, the results before and after the normalization are displayed in these plots. The plots here show the distribution of variance in groups of transcripts split by UMI on the x-axis and in percentiles of cells at these various levels of UMI based on the mean values. Before normalization, most of the variance is explained by the larger cells, while after normalization, you should see a more homogeneous distribution of variance across the percentiles of cells as well as across the various mean values of cells.